Hi. This week we'll do things a bit differently. We'll be starting our discussion or a series of discussion on various biologicals which are used in dermatology. And you may remember that are a lot of biologicals, and there are different types of biologicals. You have your TNF-alpha inhibitors. You have one or two biologicals for every interleukin, and it becomes a bit confusing and difficult to remember what all biologicals are targeting which all receptors or which all interleukins, and how are they actually leading to the clinical efficacy that we see in psoriasis. So I thought that we'll take a departure for a week. We'll first study the pathophysiology and the pathomechanisms of psoriasis so that we may know at which level of psoriatic pathology a certain cytokine or a chemokine is acting, and what are the effects and functions of that particular cytokine, so that we may know what happens when we block that cytokine. So many times you would listen or you would see in your patients that you have started one type of biological, and after a certain amount of time that biological is not working properly to its utmost extent, and you may need to switch the biological. Now we need to find out how and why psoriasis occurs. What is this complex interplay between different types of cells of the immunity, predominantly the adaptive immunity, to some extent innate immunity also, and what are the interplay of different cytokines and different kind of cell populations that secrete these cytokines, and exactly how this kind of immune mediated inflammatory dysregulation leads to a psoriatic plaque. So if we know a bit about how does psoriasis happens, which is the prototype disease for biologicals, we'll be able to easily understand at what point a certain biological acts in the pathomechanism, and how does it leads to improvement in the clinical condition of the patient who's suffering from psoriasis. So I thought this was a good way to introduce biologicals, because otherwise we'll just be that what we used to do was just mug up details and learn and just try to remember different kind of biologicals, and it becomes difficult when you don't know the background of how they act and how are they leading to improvement in patient's condition. So I thought it will be a good idea to learn a bit about pathomechanisms in this condition in psoriasis, and then using this background of pathophysiology, the background of inflammatory cascade, we can learn about each and individual biological. So this week we'll discuss just the pathomechanism of psoriasis, and we'll try to then subsequently discuss each and individual biologicals and their usage in psoriasis and other dermatoses, and how we can use them for providing that clinical benefit to our patients. So with that in mind and with that background of information, we'll start by discussing the pathophysiology of psoriasis. Now, remember that what essentially is psoriasis? It's a clinically heterogeneous disease of the skin, joint, enthesis, enthesis means tendons, and bones. So we'll deal ourselves with predominantly discussion about skin. The joints are somewhat of a similar pathology involving the synovial epithelium. So we'll discuss majorly what it does to the skin, and just a little bit correlation between other organ systems. Now, initially, psoriasis was considered as an unregulated keratinocyte proliferation, and that was quite easy to understand. Clinically, we see increased scaling in our patients, and a psoriatic plaque has a lot of scales, a lot of erythema, and on biopsies, it leads to an acanthotic epidermis, absence of granular layer, significant proliferation of the keratinocytes. And our undergraduate books mention that a normal epidermal cell turnover is roughly about 30 days. Some books mention 28 days, so 28 to 30 days. But in psoriasis, this could be as fast as 45 days. That means it is about 1/6 of the time that a normal skin takes to turn over. But this turnover leads to development of undifferentiated, or you could say not properly differentiated keratinocytes, and because of that, they are not able to get removed from the outer layers of skin and tend to stick and form scales. Okay. So initially, psoriasis was considered as a disorder of unregulated keratinocyte proliferation, but with subsequent studies, we found out that it is nothing but a chronic immune inflammatory disease. So a chronic disease which is immune mediated, and that immune mediated dysregulation is what drives the increased keratinocyte proliferation. So increased proliferation of the epidermis is a effect of the inflammatory cascade happening at the level of dermis, epidermis, and the complex interplay between different arms of immunity. So it's a complex interaction of genetic, that means in genetically predisposed individuals, not all individuals will get psoriasis by their triggers, but in genetically predisposed individuals, if we have certain kind of triggers, it might lead to start of psoriatic pathology. Triggers can be environmental and additionally with multiple factors leading to immune mediated inflammation of the skin. Okay, so multiple factors, it's a multifactorial disease and it leads to immune mediated inflammation. It is characterized as MHC1 disease, and a newer term which is coming about is MHC1opathy, and it's an umbrella term used to include all of the different pathologies which are mediated by MHC class 1. So we will not discuss that in detail, we'll just restrict ourselves to psoriasis. Now, what are the major players in psoriatic pathology? And we'll discuss some of this or nearly all of these major players in some detail. So, the majority of the pathomechanism is because of innate immune cells and predominantly the TH17 cells which secrete the interleukins 23 and 17 cytokines. Clear? Let me just draw some straighter lines. So innate immune cells, TH17 and interleukin 23 and 17 cytokines. So how does psoriasis start? There is any trigger which leads to a local inflammation, that means inflammation at the epidermal level, and this leads to breakdown of peripheral tolerance, that means the cells which are somewhat prevented from immune reaction, that prevention is taken off and the immune cells recognize these newer antigens as other antigens, and this leads to their activation. So DCs, which are dendritic cells, activate the MHC1 positive T cells, and this leads to inflammation. So the normal cascade is whenever you have an injury, this leads to dendritic cell activation, which leads to T cell activation, which leads to multiple cytokines, and this leads to inflammation. So this is just you could say a backbone of psoriasis pathogenesis. Now in the skin, the adaptive immune system plays a major role and the players are T cells, while in nail, bone and joints, the innate immune system plays a major role, and that is mediated through various cytokines which we'll be discussing further. Now I told you that certain triggers leads to local inflammation. So what are those triggers of inflammation? So it has been documented that streptococcal infection is one of the triggers in Guttate type of psoriasis, and that is, as per the recent theories, is molecular mimicry of the M protein of bacterial cell wall with keratin. That means the M protein which is present in the bacterial cell wall is somewhat antigenically similar to keratin, and when this bacteria enters our immune system, it is recognized by the T cell and it leads to inflammatory cascade to destroy that bacteria. But since the M protein is mimicking or is similar to the keratin, the T cells which are activated by those M protein will damage the keratins as collaterally because they think it's streptococcal antigen. Clear? Now the major triggers of inflammation, or the major, you could say, starting point, a very important starting point is cathelicidine LL37. Now what exactly is cathelicidine? Cathelicidine is antimicrobial peptide. These are small peptides which are secreted by the skin cells, secreted by the keratinocytes, and this leads to formation of complexes with antigens, complexes with self-antigens, predominantly DNA. And this antigen microbial peptides attaches itself. Let me just, yeah. So this antimicrobial peptide attaches itself to DNA fragments which are secreted or which are a result of keratinocyte injury. So the keratinocyte gets damaged, the cell gets damaged, it releases its DNA into circulation, and this antimicrobial peptide also by the keratinocytes, they both come together to form a complex, and this complex activates dendritic cells via toll-like receptor 9. And when the dendritic cells are activated, it will lead to an inflammatory cascade. Okay. Now, so we have heard about cathelicidin activating dendritic cells. So let's go to the next step. So these are the dendritic cells, and you will hear about a lot of dendritic cells in psoriatic pathology. You will hear about plasmacytoid. You will hear about inflammatory. You will hear about myeloid lineage of dendritic cells. So we'll discuss them first and foremost. We're going to discuss plasmacytoid dendritic cells, which are the major mediators of inflammation in psoriasis. Now, these dendritic cells denote an activated phenotype with the expression of CD80, 83, and 86. And when these cells are activated, they will secrete the cytokines and chemokines interferon alpha, interleukin 12, interleukin 23. So, activated dendritic cells will secrete interleukin 23. And in the presence of these cytokines, the T cells will convert into a TH1 phenotype. Now remember that there was initially considered to be two different phenotypes. You have TH1 and TH2. TH1 used to deal with intracellular pathogens, TH2 by extracellular pathogens. But with increasing study, we have found another pathway which is TH17. And TH17 is found to be the most important pathway in psoriasis as per the current theories. Okay. Now this inflammatory dendritic cells which have been activated by cathelicidin secrete interleukin 1, interleukin 6 and transforming growth factor beta and interleukin 23. Now this interleukin 23 is one of the major cytokine, the major driver of inflammation as far as cytokines are concerned. And in the presence of interleukin 23, the T cells form a subset of T cells known as TH17 cells. And these TH17 cells are going to secrete cytokines interleukin 17A and interleukin 22, and these are the downstream cytokines. Downstream cytokines for psoriatic inflammation. Okay. Now, remember that initially psoriasis was considered as TH1 disease, but now it has been found to be predominantly mediated by TH17. Okay. Studies show that TH1 cells gets activated in the same manner as TH17 cells are getting activated. That means inflammatory dendritic cells are leading to development of TH1 cells. Now TH1 cells will secrete interferon gamma, interleukin 2 and TNF-alpha, and this will lead to perpetuation of inflammation, and thus the disease becomes chronic. Okay, so these cytokines secreted by the TH1 subset of T cells will lead to perpetuation of inflammation, making the disease chronic. The TH17 cells are the most dominant cell population seen in a psoriatic lesion, and these are the major players. So there will be inflammation leading to cathelicidin, leading to activation of dendritic cells, leading to activation of T cells which form into TH17 and TH1. TH1 is predominantly responsible for chronicity of disease, and TH17 is responsible for secretion of various kinds of cytokines which will further lead to the clinical manifestation of psoriasis. Clear at that? So, we have discussed about dendritic cells, and dendritic cells leads to activation of T cells. Now we'll study a little bit about T cells. Now T cells are of two types, CD8 and CD4. The activated CD8 T cells migrate from the vessels to the epidermis. Now whenever dendritic cells, these dendritic cells, when they are activated, they will go to the nearest draining lymph node, and this lymph node, they will express these antigens. We all know that dendritic cells are a group of cells in antigen presenting cells. Now these dendritic cells will present an antigen. Remember that there is no such particular antigen, but dendritic cells are recognizing self-antigens as other antigens, and they are presenting these antigens to T cells. Okay. So this dendritic cell will present an antigen in the lymph node to T cells, and these T cells will form different subsets. Now in dermis, CD4 positive T cells are the predominant cell population, while in the epidermis, CD8 positive cells are predominant. Bologna also mentioned that in the dermis, CD8 positive cells are also seen. There's a mixture, but the cell population in dermis is predominantly CD4 type. Clear? Now these T cells which are activated in the lymph node, they will express various homing antigens. Homing antigens are like CTLA and addressing, and when any T cell will express skin homing antigens, that is a marker that these cells will go to the skin now. Okay, and because of these cells, because of these markers, it will go to the skin and it will lead to inflammation and thus psoriasis. Okay. Now there are other cell populations also which are also activated by dendritic cells, and these cells have predominant function of secreting various cytokines and chemokines with nearly similar function as those cytokines secreted by other T cells, and this leads to increased inflammation. So the action is same as other T cells, it is just there are different cell population. One of them is natural killer cells, and natural killer cells have been found to be around 5% to 8% in the cellular infiltrate of psoriasis, and their predominant location is at mid and papillary dermis. Clear? Now these NK cells have a lower CD57 expression, and thus they are of the immature phenotype. They have not matured enough. And because of this immature phenotype, they produce more cytokines, predominantly interferon gamma. Okay, and they have a heightened response to interleukin 2 stimulation. They have higher degranulation capacity and a greater turnover. That means they will multiply a lot. They will have a lot of number, a lot of an increased population, and they are going to secrete a lot of cytokines because they are immature. They don't know what they are doing. Okay. Now, what is the role of keratinocyte? As we have previously discussed that initially psoriasis was thought as a disorder of keratinocyte proliferation. Then with further studies we found that it is predominantly a T cell mediated disorder, and that inflammatory disorder is leading to proliferation of keratinocytes. So it was found that keratinocytes was more or less like an effect. Okay, they don't take part in inflammation, but because of inflammatory cascades happening in different subsets of T cells, they are multiplying. But further study shows that that is not the case. It's not like keratinocytes don't contribute to the inflammatory cascade in psoriasis. It's just that they do contribute a bit differently. For example, we have already heard about cathelicidin. Okay, and this is found when the keratinocytes are destroyed. Okay. Now, Keratin-17 is one of the keratin, and it has been, in some studies, has been implicated as an autoantigen for psoriasis. So remember when I said that dendritic cells were presenting some antigens to the T cells in lymph node, one of the antigen has been speculated to be Keratin-17. There might be multiple other antigens also. Now we will study a little bit about DAMP and AMP. AMP is antimicrobial peptide. We have studied a bit about in previous slides, and DAMP is just damage associated microbial proteins. Okay, so what happens is that whenever there is a cell and it gets infected by a bacteria or a pathogen, it will kill itself. To prevent the infection, it will kill itself, and it will secrete a lot of chemicals. When the cell dies, a lot of chemicals will be outside the cell, and these chemicals act as a trigger for inflammation. It's like the cell is saying that there is something wrong in the body, something has infected me, I am dying, but please make note of my products so that you will know that something is wrong, and you should start an inflammatory cascade to destroy that organism which is infecting me. Okay, so these cells get damaged, and that is why it is damage associated microbial peptides. And these peptides are predominantly self DNA, that means body's own DNA, or self RNA, that is body's own RNA. Okay. Antimicrobial peptides are also found in keratinocytes inside these cells, And these include beta-defensin, S100, and cathelicidine. Cathelicidine is, we have discussed previously, one of the important antimicrobial peptides. Now, secretion of these antimicrobial peptides will activate the keratinocytes, and keratinocytes will itself secrete a lot of homing homing chemokines. Now, what happens is that whenever keratinocyte is undergoing any kind of injury, it will start to secrete some chemicals to call T cells towards it. So when T cells will come, they will see that the keratinocyte is injured, and they will start the process of clearing the infection or clearing the trigger and leading to cell recovery. That happens in the normal process. But in psoriasis, the T cells are activated and they will come, and since they are activated by autoantigens, they will find normal antigens, normal molecules. They will think these are other antigens or non-self antigens, and this will lead to an increased inflammation at site. And that is why it has been implicated that trauma leads to development of psoriatic plaque, which we call as coblerization. So this is one of the theories behind the presence of coblerization in psoriasis. Cathelicidine LL37, which is the complete name, also leads to vascularization and vasodilation. What it does is that since the vessels are dilated, there will be more transfer of activated T cells at the site and more inflammation. And this is the reason, or one of the reasons, there are a lot of angiogenic molecules secreted in psoriasis. One of the reasons that we see dilated and tortuous capillaries in psoriatic biopsies. Other proteins like ADAMTS, like protein 5 from injured melanocytes, have also been implicated in psoriasis. Additionally, there is a STAT3 upregulation. So we'll discuss STAT3 in the future when we'll be discussing JAK-STAT inhibitors. But STAT3 upregulation leads to transforming growth factor alpha secretion, and this acts as increased proliferation of keratinocytes. Okay, so you can see that the STAT3 upregulation leads to secretion of TGF-alpha, and this leads to secretion of increased proliferation of keratinocytes. Now we have understood that keratinocytes can also act as a start of inflammation and also as perpetuator of inflammation or increasing inflammation. And as the result of this increased inflammation, it will lead to increased proliferation of keratinocytes and thus the psoriatic plaque. We are clear on that? So we have learnt a bit about skin damage leading to dendritic cells activation, leading to T cell activation, leading to secretion of various cytokines and chemokines. So very quickly, we'll just go through a list of cytokines and chemokines and their function in psoriasis. Clear? First one is interferon alpha. It is secreted by plasma cytodendritic cells and it has been found to be increased in psoriatic skin. Clear? Other important cytokine is TNF-alpha. Now, TNF-alpha is secreted by activated dendritic cells, the TH17 cells and TH1 cells. It is the first and the foremost very important inflammatory mediator. In the presence of TNF-alpha, there is additionally, so activated dendritic cells will secrete a lot of cytokines and chemokines. One of them is TNF-alpha. Another one of them is interleukin 23. What TNF-alpha does is it increases the inflammation. What interleukin 23 does is it will lead to formation of TH17 cells. Okay, so proliferation and survival of TH17 cells. Interleukin 23 is produced by myeloid dendritic cells and at low levels by keratinocytes also on toll-like receptor stimulation. So the sources of interleukin 23 is myeloid dendritic cells, which are the major source, and some minor secretion by keratinocytes on toll-like receptor stimulation. The next cytokine which we'll study is interleukin 17. Okay, so TNF-alpha by dendritic cells, 23 by dendritic cells, and because of 23 there is increased proliferation of TH17 cells, and these TH17 cells will produce interleukin 17A. So these are produced by TH17 cells. They are elevated skin lesions and they are found to be in serum of psoriasis. It leads to activation and recruitment of neutrophils. Remember that in biopsies of psoriasis we see neutrophilic microabscesses. They go by the name of Pogos or micromonroe abscess. So we see a lot of neutrophils collected inside the epidermis. And why are the neutrophils there? Because they are being called by different cytokines, namely interleukin 17, which is one of those cytokines. Additionally, there's enhancement of angiogenesis, that means more blood vessels are being formed, and promotion of the release of other inflammatory cytokines like TNF-alpha. So remember that interleukin 17 leads to formation of more TNF-alpha, and this is a positive feedback loop, and thus the inflammation cascade is going to increase with time. Interleukin 17A can also directly activate keratinocytes to force them or even to stimulate them to secrete different cytokines which have been implicated in pathway. So now we have reached the level of cytokines and chemokines. We started with injury leading to amps and dams. Then we went to dendritic cells. Then we went to activated T cells of TH1 and TH17, and now we are dealing with cytokines and chemokines secreted by those T cells. Okay. Other interleukins are like interleukin 12. They are by the activated myeloid dendritic cells, and these are responsible for TH1 differentiation. And TH1 cells will secrete interferon gamma. Interleukin 22 is secreted by 17 and 22 helper T cells, and with interleukin 17, it forms what is known as interleukin 17 interleukin 22 axis, which is one of the important inflammatory axes in psoriasis, and we'll just discuss that in brief in subsequent slides. Okay. Now, interleukin 22 is secreted by TH17 and TH22. It leads to growth and activation of keratinocyte by inducing the STAT3 pathway. So remember in previous slides we said that STAT3 is upregulated, and this upregulation with the usage of transforming growth factor alpha leads to hyperproliferation of keratinocytes. Okay, so STAT3 activation leads to hyperproliferation. The anti-macropeptides also increase, and it also increases matrix metalloproteinases. Interleukin 20 is also one of the interleukins in IL-20 family. They are produced by keratinocyte and have a synergistic role like interleukin 22, somewhat similar role. And this leads to thickness and epidermal thickness and maturation, which we see in biopsy as acanthosis. Clear? Interleukin 36. Now, interleukin 36 have been implicated, or rather its receptor and different types of interleukin 36 have been implicated in majorly pustular psoriasis. Clear? So, in pustular psoriasis, IL-36 play a very major role, and also remember that the action is similar to interleukin 1, but only skin is affected, and that is why in DITRA, which is deficiency of interleukin 36 receptor agonist, the clinical effect is seen only in skin, while in DIRA, other organ systems like joints are also affected. So different manifestation of different organ systems are seen in DIRA, but in DITRA, we just see the skin manifestation. Additional cytokines are like vascular endothelial growth factors, which is responsible for increased vascular permeability, and we see that in the form of increased redness, erythema, increased chemotaxis of inflammatory cells at the skin site. Clear? So we started with trigger, cathelicidine, dendritic cells, T cells, cytokines and chemokines. So we have understood a bit of the pathway of psoriasis. Okay. So just to recapitulate, any injury or any trigger of inflammation to keratinocytes will lead to secretion of beta defensins, cathelicidine LLG7, and also exposure of self-DNA to immune cells. Clear? This will lead to activation of plasmacytoid dendritic cells. So this is dendritic cell. Activation of plasmacytoid dendritic cell through toll-like receptor 9 will secrete interferon alpha. Clear? One pathway is clear. Second is that same cathelicidine LL37 will bind to self-RNA and self-DNA and lead to myeloid dendritic cell maturation. And when myeloid dendritic cells get mature, they will secrete a number of cytokines and chemokines, namely TNF-alpha 23, 12, and 6. Now, interferon alpha is one of the major mediator. It is the first and the major mediator of inflammation. Okay, followed by interleukin 23, and the next major mediator is interleukin 17. Clear? Now what interleukin 23 does is, here is interleukin 23. Interleukin 23 under the exposure of TGF beta, interleukin 6 and interleukin 1 beta will lead to conversion of naive T cell to 17 subset of T cell. So naive T cell will get converted to TH17 cells, and this is because of interleukin 23. And this TH17 cell will secrete interleukin 17, interleukin 22 and TNF-alpha. So see that TNF-alpha is secreted by myeloid dendritic cells, which also secrete 23. 23 will lead to TH17 and 17 will lead to more TNF-alpha. So TNF-alpha keeps on increasing. Okay, this inflammatory marker keeps on increasing, and that is why TNF-alpha inhibitors are one of the major mainstay biologicals used to treat psoriasis. Clear? Additionally, there are various interleukins like 23, 17, here is 12, 22. So blockage of these individual interleukin by various different kinds of biologicals leads to clinical efficacy. So we know at what aspect these interleukins are acting, and then we will know what all to block with different types of biologicals available. Okay. Additionally, interleukin 23 leads to FOXP3 downregulation, and FOXP3 is responsible for good development of T regulatory cells. So when FOXP3 is downregulated, the T regulatory cells are also downregulated. That means at one point you are having an increased activation of immune T cells, namely TH1 and TH17, and there is decrease in regulatory T cells. So the inflammatory cascade keeps on increasing and without any breaks, without any stoppage, and this leads to inflammatory activity in psoriasis. I hope I've made myself clear how do different kind of interplay between T cells and their cytokines and chemokines leads to development of psoriasis. Clear? So now let's discuss one or two cytokines which are important. So TNF-alpha, as I have told you, is one of the most important cytokines implicated in psoriasis, and additionally one of the major cytokines. It has been found to be upregulated in psoriatic plaque, and the level of TNF-alpha in serum and the lesion, if this is predominantly seen in lesion, correlates with the severity of psoriasis. It is secreted by CD11C dermal dendritic cells and CD163 macrophages. It also upregulates the intracellular adhesion molecules on the surface of endothelial cells. Now, we have endothelial cell lining of our capillaries, and if adhesion molecules are increased because of TNF-alpha, more inflammatory cells will get attached itself at the endothelial cells, and when T cells get attached to endothelial cells, they will migrate in between the gaps and will lead to inflammation at the epidermal site. TNF-alpha is also chemotactic for neutrophils, and that is why you see neutrophilic microabscesses. TNF-alpha additionally activates dendritic cells. That means activated dendritic cells secrete TNF-alpha, and TNF-alpha additionally activates dendritic cells. Again, a positive loop increasing TNF-alpha. Clear? In the last slide, we have discussed that increased activation of dendritic cell will lead to increased formation or increased secretion of interleukin 23, and there will be increased activation of TH17, and TH17 will itself lead to increased TNF-alpha. So now you see how TNF-alpha is increasing itself, how TNF-alpha is increasing TH17, and how TH17 is increasing TNF-alpha. Clear? TNF-alpha also leads to keratinocyte proliferation, so cells are getting thicker, and also stimulates interleukin 22 receptor. And in the previous slide, we have read about that in TH17 cells secrete IL-17 and IL-22, and IL-22 receptor is also activated by TNF-alpha. TNF-alpha by itself can also activate NFK-beta pathway. And remember that when NFK-beta gets activated, it will lead to increased translation of inflammatory cytokines. So remember on our previous videos on drugs like cyclosporin and glucocorticoids, if you have a genetic material and NFK-beta gets activated, it will lead to attaching itself to response elements on the genetic material, and this attachment will lead to increased translation of that particular segment of genetic material which encodes for certain cytokines. and this will lead to increased inflammatory cytokines and further increasing inflammation. So now we have read a bit about TNF-alpha. Clear? The next cytokine is interleukin-17A and interleukin-22, which are secreted by the TH17 subgroup of T cells. Interleukin-22 has been found to correlate well with disease severity. Both of these interleukin leads to keratinocyte proliferation. They also express markers like CXCL1, 2 and 8, and these markers are additionally neutrophilic chemoattractant. So along with TNF-alpha, interleukin-17A and interleukin-22 will also lead to migration of neutrophils from the blood vessels to the epidermis, forming microabscess. Now CCL20 is a ligand. So in this image you can see this orange ball is CCL20 and it is secreted by keratinocytes. It attaches itself to CCR6. CCR6 is the receptor for this ligand. So you can remember that R is receptor, L is ligand. So CCR6 positive TH17 cells. So these are 17 cells which are CCR6 positive. When CCL20 attaches itself, leads to activation of TH17 cell. And it's a homing molecule, and that is why the epidermis, the keratinocyte will attract this activated TH cells to the epidermis. Clear? And because of that, you will see T cell migration at the epidermal level. Additionally, interleukin-17 will activate NFK beta, and like we discussed in the last slide, increased activation of NFK beta will lead to increased inflammatory cytokine translation, further inflammation. Now, this activation of CCR6 with CCL20 secreted by keratinocyte forms an important axis known as CCR6-CCL20 axis, and this is also one of the important axes. Now, TNF-alpha leads to TNF-alpha interleukin-17A interleukin-23 axis, and this is the most important axis. So remember, don't get confused. Interleukin-17-23 axis is the most important axis in psoriatic pathology. And additionally, current studies say that CCR6 and activation by CCL20 attachment is also one of the important axes. But if you have to answer one, the answer would be this. This is just a recent theory regarding how keratinocytes take part in the inflammatory cascade. So just a little slide on CCL20, CCR6. CCL20 is constantly expressed in keratinocytes, which are the major source of CCL20, and it is upregulated by TNF-alpha and interleukin-17A and interleukin-23. Additionally, human keratinocytes produce beta-defensin 2, which is a non-chemokine ligand for CCR6. So, you have a T cell and you have a receptor known as CCR6 on it. And these are keratinocytes. Now, keratinocytes secrete a ligand known as CCL20, and this CCL20 attaches itself to CCR6, and this attachment will ask the T cell to come to the epidermal site and lead to inflammation. Clear? Additionally, they also secrete beta-defensin 2, and it has the same action as CCL20. The only difference is that CCL20 is a chemokine, beta-defensin 2 is an antimicrobial peptide. Clear? And the function of these two is to attract TH17 cells to the epidermis where the inflammation will take place. Clear? Now CCL20 CCR6 axis, in this axis CCL20 has been regarded as an alarm mediator for keratinocyte injury. That means whenever keratinocytes are injured, they will secrete CCL20 like an alarm, and this will lead to migration of TH17 cells to the milieu, and this is what is known as one of the reasons of covernalization or development of psoriasis, at trauma sites. Okay, CCL20 and CCR6 axis is also implicated in psoriatic arthritis. Okay, now they have been found that in keratinocyte, the keratinocyte also express CCR6, but the function is not known as of now. Additionally, CCL20 is reduced by biologicals like infliximab and adalimumab. So certain biologicals, in spite of their action on TNF blockade, will also lead to decreasing CCL20 and thus decreasing the activation of TH17. So that is an additional function of biologicals. But we'll discuss this function in detail when we are discussing individual biologicals. Clear? Now, I want all of you to just for a few seconds just look at it. Do not understand what is happening. Do not look at the arrows. Just take this picture inside your mind and just look at the picture. Do not look at the legends. And slowly we'll just summarize the pathomechanism of psoriasis and how what all we have studied previously, all the T cells, all the interleukins, how are they leading to psoriatic manifestations. Clear? So let's start. This is the initiation phase and this is the pathological phase. So initiation happens by any trigger which will lead to damage of the keratinocytes. Now this keratinocytes get stressed, they're going to secrete a lot of antimicrobes and damage associated proteins. Additionally, self-DNA, self-RNA gets secreted into the circulation when the cell gets damaged. Okay? And this happens in all individuals. But whether the immune system will react to it or not is decided by the genetic makeup of the individual. So in genetically predisposed individuals who carry the susceptibility alleles of psoriasis genes will result in disease formation as an effect of this triggering damage to keratinocytes. Clear? Now this will lead, this antimicrobial peptide, this green box is LL37 cathelicidine. Okay. Now cathelicidine will attach itself to self DNA and self RNA, and this attached complex will further activate plasmacytoid dendritic cells. When this plasmacytoid dendritic cell gets activated, okay, this will lead to activation of the dermal dendritic cells. Clear? Now, when these dermal dendritic cells get activated, these dermal dendritic cells will migrate to the nearest lymph node, and in the lymph node, they will present these complexes to naive T cells. And these naive T cells, under the action of various cytokines secreted by these cells, dendritic cells, will mature into different type of subsets of T cells. Namely, three subsets have been implicated: TH1 subset, TH17 subset, and TH22 subset. So these three subsets have been implicated in lymph node under the activation by an activated dendritic cells. Clear? Now these dendritic cells can be individually and separately activated by damaged keratinocytes by various cytokines and chemokines. Okay. Now, when these T cells get activated by these dendritic cells, they will go into the circulation into the blood vessels. Clear? Now, because of increased expression of these lectin molecules, predominantly by the action of TNF-alpha and other cytokines, they will migrate out of these blood vessels and will reach the dermal epidermal junction and the epidermis, where the inflammation is supposed to happen. Am I clear in that? Okay. And with that, what happens is that TH17 cells, so let's just take them one by one. TH1 cells will lead to TH1 cytokines, which are normally interferon alpha and interferon gamma, and this will attack the keratinocytes for further destruction of keratinocytes. And this keratinocyte destruction and also increased proliferation of keratinocytes leads to increased epidermal turnover, leading to the psoriatic plaque. Clear? And additionally, remember in previous slides we discussed that interferon gamma is also responsible for chronicity of disease. So because of TH1 action, the disease is a chronic disease. Okay? TH17 is going to secrete the major interleukin player, which is interleukin 17, which will lead to inflammatory cascade and inflammation, leading to a development of psoriatic plaque. Clear? So TH1 cells will release interferon gamma and TNF-alpha. This will amplify the inflammatory cascade and they will act on keratinocyte and lead to chronicity of disease. The TH17 cells will secrete interleukin 17A and this will stimulate the keratinocyte proliferation like we have discussed previously, and also release of various antimicrobial peptides which will further keep on increasing the inflammatory cascade. Now these are the TH22 which will secrete interleukin 22 with similar action as the interleukin 17. It has the same action, and all of these cytokines and chemokines will also attract various neutrophils. So neutrophils will also come from blood vessels and form microabscesses here. Okay, so these microabscesses are found here and this will lead to micromonro abscess or Kogo's abscess. So clear? So I hope we have been able to understand how the psoriasis develop. A trigger, or if I just point it out here, a trigger leads to release of various, let me just change the color so that it's easily. A trigger leads to keratinocyte injury. Keratinocyte injury will lead to release of various self-antigens which attaches itself to cathelicidin. These attached complexes will then activate plasmacytoid dendritic cells. These plasmacytoid dendritic cells then will activate dermal dendritic cells. These activated dermal dendritic cells will go in lymph node. In lymph node, they will convert naive T cells into three subsets: TH1, TH17, and TH22. These three subsets will migrate from the blood vessels to the epidermis. TH1 will release interferon gamma and TNF-alpha, leading to inflammatory cascade and epidermal cell turnover and keratinocyte proliferation. Same thing will be done by interleukin 17 and same thing will be done by interleukin 22, and this leads to development of psoriatic plaque. Okay, so I hope I have been able to make this pathomechanism of psoriasis clear, and you will be able to answer good in your vivas or maybe write a good short note on the pathomechanism of psoriasis. We have not gone into depth regarding various triggers like environmental factors or infections, or we have not gone into the genetic basis of psoriasis. That's a different lecture altogether, and let me know if you want that. I have my notes ready so I can make a video on the genetic, all the genes which have been implicated, SOR1 to SOR6. They have been implicated and we can have a separate video on that. But it is important for us to understand the pathomechanism of psoriasis, because once we know how psoriasis develops, we will know how different biologicals act. Okay, and I hope this kind of approach to biologicals will make understanding them easier, and and then we'll be able to decide what all biologicals we are going to start. So remember that there is no one biological to treat psoriasis. It is filled, it has a plethora of different biologicals which can be used, and every biological is different. So you may want to decide using infliximab or adalimumab or ustekinumab or guselkumab or secukinumab. So all of these biologicals can be used, but depending on what separately you are targeting, every biological has different efficacy in psoriasis, and in subsequent from next week onwards, we'll be starting to discuss each and individual biological and how does it leads to efficacy in psoriasis and what are the other uses in different other kind of skin disorders, and we will treat each biological like a drug, and we'll come back to our discussion on one drug every week in biologic. If the videos are shorter, I may decide to do 2 biologicals in a week, depending on that. Till then, if there are any doubts, any suggestions, any queries, you can just comment below the video, or you can email me directly on my email ID, or you can message me on WhatsApp and everything. And let me know what if you need any clarification regarding pathomechanism of psoriasis, or if you need any other any doubts you have, or any other contribution, something that I should have covered but I'm not covered, just let me know. Okay, and with that I'll just end this presentation, and we'll start from biologicals next week, and do it as we have been doing previous drugs every week. Okay, so with that the presentation finishes. Adios, bye bye.